Alrighty, and hello again. Welcome to another chapter of Advanced Pathophysiology. Today we're jumping into chapter 35, which is our structure and function of the pulmonary system. So, my pages, as always, will not be lining up as yours do, as I'm using a PDF. However, we'll get there all the same. So, Chapter 35, the structure and function of the pulmonary system. I believe we'll be reading the entirety of the chapter, so we'll jump right into it. Again, my pages may not match up to yours, but that shouldn't matter. So, the primary function of the pulmonary system is the exchange of gases between the environmental air and the blood. There are three steps in this process: ventilation, the movement of air in and out of the lungs; diffusion, the movement of the gases between the air space in the lungs and the bloodstream; and perfusion, the movement of the blood into and out of the capillary beds of the lungs to the body organs and tissues. The first two functions are carried out by the pulmonary system, the third via the cardiovascular system. Normally, the pulmonary system functions efficiently under a variety of conditions and with little energy expenditure. So, what are our structures of the pulmonary system? The pulmonary system includes two lungs, the upper and the lower airway, and the blood vessels that serve them, the chest wall, a thoracic cage, and a diaphragm. The lungs are divided into lobes, the three on the right and the two on the left. Each lobe is further divided into segments and lobules. The mediastinum is the space between the lungs and contains the heart, the great vessels, and esophagus. A set of conducting airways or bronchi delivers air into each section of the lung. The lung tissue that surrounds the airways supports them, preventing their distortion or collapse as gas moves in and out during ventilation. The diaphragm is a dome-shaped muscle that separates the thoracic and abdominal cavity and is involved in ventilation. So, looking at the structural plan of the respiratory system, the inset shows alveolar sacs, where the interchange of oxygen and carbon dioxide takes place through the walls of the grape-like alveoli, the capillaries surrounding the alveoli. And we see that here in the picture. Also, the upper respiratory tract and the lower respiratory tract. The lungs are protected from a variety of exogenous contaminants by a series of mechanical and cellular defenses. So I'm just going to make this a little bigger. Boom. These defense mechanisms are so effective that a healthy individual, I'll repeat that. The defense mechanisms are so effective that in the healthy individual, contamination of the lungs itself, particularly by infective agents, is relatively rare. So, a list of ways that the body will protect itself: um, upper respiratory tract mucosa, the hair on your nose, your branching airways, your mucus blanket, the innate immune protections or proteins, your cilia, alveolar macrophages, our surfactant, your irritant receptors in the nares in your nostrils, and your irritant receptors in the trachea and large airways. So your conducting airways. The conducting airways provide a passage for the movement of the air into and out of the gas exchange structure of the lungs. The nasopharynx, oropharynx, and related structures are called the upper airway. These structures aligned with ciliated mucosa, with a rich vascular supply that warms and humidifies inspired air and removes foreign particles from it as it passes into the lungs. The mouth and the oropharynx provide for ventilation when the nose is obstructed or when increased flow is required. For example, during exercise, filtering and humidifying are not as effective and efficient than with mouth breathing. The larynx connects the upper and lower airways and consists of the endolarynx, and its surrounding triangular-shaped bony and cartilaginous structure. The endolarynx is formed by two pairs of folds, the false vocal cords, or supraglottis, and the true vocal cords. The slit-shaped space between the true cords forms the glottis. The vestibule is a space above the false vocal cords. The laryngeal box is formed by three large cartilages, the epiglottis, the thyroid, and the cricoid, and the three smaller cartilages, the arytenoid, the corniculate, and the cuneiform, connected by ligaments. The supporting cartilages prevent collapse of the larynx during inspiration and swallowing. The internal laryngeal muscles control vocal cord length and tension, and the external laryngeal muscles move the larynx as a whole. Both sets of muscles are important to swallowing, respiration, and vocalization. The internal muscles contract during swallowing to prevent aspiration into the trachea and contribute to voice pitch. The trachea, which is supported by U-shaped cartilage, connects the larynx to the bronchi, the conducting airways of the lungs. The trachea divides into the two main airways or bronchi at the carina. This area is very sensitive and when stimulated can cause coughing and airway narrowing. The left main stem, bronchus, branch from the trachea is about a 45-degree angle. The right mainstem bronchus is slightly larger and more vertical than the left, and branches at about a 20 to 30-degree angle from the trachea. Aspirated fluids, or foreign particles, tend to enter the right lung rather than the left. The right and left main bronchi enter the lungs at the hila, or root of the lungs, along with the pulmonary blood and lymphatic vessels. From the hila, the main bronchi branch into lobar bronchi, and then to segmental and subsegmental bronchi, and finally end at the 16th division in the smallest of the conducting airways, the terminal bronchioles. With the multiple divisions, the cross-section area of the airways increases to 20 times that of the trachea. This results in decreased velocity of airflow into the gas exchange portion of the lung and allows for optimal gas diffusion. So again, we see the separation in the conducting airway from the trachea, the segmental bronchi, and then your subsegmental bronchi, your non-respiratory and respiratory, to the alveolar duct. The bottom portion here also shows how the ciliated cell mucus layers change, where we have smooth muscle and basal cells, your lamina propria and basement membrane. Finally, your Pleura cells and your nerves. The bronchial walls have three layers: the epithelial lining, a smooth muscle layer, and connective tissue layer. In the large bronchi, up to approximately the 10th division, the connective tissue layer contains cartilage. The epithelial lining of the bronchi contains single-celled exocrine glands, the mucus-secreting goblet cells, and ciliated cells. Higher columnar pseudostratified epithelium lines the larger airways and becomes progressively thinner, changing to columnar cuboidal epithelium in the bronchioles and squamous epithelium in the alveoli. The submucosal glands of the bronchial lining produce a mucus blanket that protects the bronchial epithelium. The ciliated epithelial cells rhythmically beat this mucus blanket towards the trachea and pharynx, where it can be swallowed or coughed out. Towards the terminal bronchioles, ciliated cells and goblet cells become more sparse, and smooth muscle and connective tissue layers begin to thin. So what is that gas exchange airway? The conducting airways terminate in the respiratory, the terminal bronchioles, the alveolar ducts, and alveoli, or alveolus, singular. These thin-walled structures participate in gas exchange, and the clusters of alveoli are sometimes called acinus. The bronchioles from the 16th through the 23rd division contain increasing number of alveoli and are called the respiratory bronchioles. The walls of the respiratory bronchioles are very thin, consisting of an epithelial layer devoid of cilia and goblet cells, very little smooth muscle fiber, and very thin and elastic connective tissue layers. These bronchioles end in alveolar ducts, which lead to the alveolar sacs made up of numerous alveoli. The alveoli are primary gas exchange units of the lungs, where oxygen enters the blood and CO2 is removed. Tiny passages, called pores of Kohn, permit some air to pass through the septa from the alveolus to alveolus, promoting collateral ventilation and even distribution of air among the alveoli. The lungs contain approximately 50 million alveoli at birth and about 480 million by adulthood. So here we're taking a look at the alveoli. The bronchioles subdivide to form tiny tubes called alveolar ducts that end in clusters of alveoli called alveolar sacs. The lung epithelial cells provide a protective interface with the environment and are essential for adequate gas exchange, preventing entry of foreign agents, regulating ion and water transport, and maintaining mechanical stability of the alveoli. The alveolar septa consists of an epithelial layer and a thin elastic basement membrane, but no muscle layer. The two major types of epithelial cells, pneumocytes, appear in the alveolus. Your type 1 alveolar cells provide structure, and type 2 alveolar cells secrete surfactant. Surfactant is a lipoprotein that coats the inner surface of the alveolus and facilitates its expansion during inspiration, which lowers alveolar surface tension at the end of expiration, thereby preventing that lung from collapsing or having atelectasis. Surfactant proteins contribute to the control of lung inflammation by decreasing the release of pro-inflammatory mediators, preventing oxidative injury, and regulating the role of fibroblasts in the airway remodeling. Surfactant proteins also are the bacteriostatic and function as opsonins and presenting pathogens to alveolar macrophages. The macrophages are the most numerous immune cells present in the lung environment and provide innate immune defense of the airway from the bronchi to the alveoli. In the alveoli, alveolar macrophages provide protection by clearing surfactant from the lung and ingesting foreign material and pathogens that reach the alveolus, preparing these substances for removal through the lymphatics. Your phagocytosis and the mononuclear phagocyte system that was described in chapters 7 and 8. Surfactant and alveolar macrophages work together with the normal pulmonary microbiota to prevent lower lung infection. Changes in the pulmonary microbiome are associated with many pulmonary diseases. Your pulmonary and bronchial circulation. The pulmonary circulation provides an extensive surface area for gas exchange, delivers nutrients to lung tissues, acts as a blood reservoir for the left ventricle, and serves as a filtering system that removes clots, air, and other debris from the circulation. The pulmonary vasculature is composed of three compartments connected in series: your arteries, your capillaries, and your veins. Looking at this picture here, we have the pulmonary circulation. The right and left pulmonary veins and arteries and the branching capillaries are illustrated. Note the pulmonary arteries carry venous blood and the pulmonary veins carry arterial blood, one of the few times in the body where things are flipped. Although the entire cardiac output from the right ventricle goes into the lungs, the pulmonary circulation has a lower pressure and resistance than the systemic circulation. Pulmonary arteries are exposed to about 1/5 of the pressure of the systemic circulation and have a much thinner muscle layer. Mean pulmonary arterial pressure is 18 milligrams of mercury. Mean aortic pressure is 90 milligrams of mercury. About 1/3 of the pulmonary vessels are filled with blood or perfused at any given time. More vessels become perfused when the right ventricular cardiac output increases. Therefore, increased delivery of blood to the lungs does not normally increase mean pulmonary arterial pressure. During exercise, pulmonary arterial pressure will normally increase, but not to more than 30 milligrams of mercury and is age and sex dependent. Numbers that you should remember is that your mean pulmonary pressure in the artery, or the mean pulmonary artery pressure, is 18 during rest, and it is not more than 30 with exercise. The pulmonary artery divides and enters the lung at the hilus, branching with each main bronchus, and with the bronchi at every division. Thus, every bronchus and bronchiole has an accompanying artery and or arteriole. The arterioles divide at the terminal bronchiole to form a network of pulmonary capillaries around the acinus. Capillary walls consist of an endothelial layer and a thin basement membrane, which often fuses with the basement membrane of the alveolar septum. The shared alveolar and capillary bonds compose the alveolo-capillary membrane, a very thin membrane made up of the alveolar epithelium, the alveolar basement membrane, an interstitial space, the capillary basement membrane, and the capillary endothelium. There's only about a 0.2 nanoseparation between blood in the capillaries and gas of the alveolus. Gas exchange occurs across the alveolo-capillary membrane. These extremely thin alveolar walls are easily damaged and can leak plasma and blood into the alveolar space. With normal perfusion, about 100 ml of blood in the pulmonary capillary bed is spread very thinly over about 70 to 100 meters squared of alveolar surface area. Any disorder that thickens the membrane impairs gas exchange, such as fibrotic lung disease. Looking at the picture here, a cross-section through the alveolus showing histology of the alveolar capillary membrane. The dense network of capillaries forms an almost continuous sheet of blood in the alveolar walls, providing a very efficient arrangement for gas exchange. So each pulmonary vein drains several pulmonary capillaries. Unlike the pulmonary arteries, which follow the branching bronchi, pulmonary veins are dispersed randomly throughout the lungs, and then leave the lung at the hyla and enter the left atrium. They are similar to the veins in the systemic circulation, but they have no valves. The bronchial circulation is part of the systemic circulation and receive 1% of cardiac output. The bronchial arteries supply blood to the trachea, bronchi, and its branches, the esophagus, visceral pleura, and vasa vasorum of the thoracic aorta, and the pulmonary arteries and to the nerves, pulmonary veins, and lymph nodes in the thorax. Not all of the capillaries drain in their own venous system. Some empty into the pulmonary vein and contribute to the normal venous admixture of oxygenated and deoxygenated to the normal venous blood. Right to left, shunting. The bronchial circulation does not participate in gas exchange. Lung vasculature also includes deep and superficial pulmonary lymphatic capillaries. The deep lymphatic capillaries begin at the level of the terminal bronchioles. Fluid and alveolar macrophages migrate from the alveoli to the terminal bronchioles, where they enter the lymphatic system. The superficial lymphatic capillaries drain the membrane that surrounds the lungs. Both deep and superficial lymphatic vessels leave the lung at the hilum through a series of mediastinal lymph nodes. The lymphatic system plays an important role in keeping the lungs free of fluid and providing an immune defense. Control of the pulmonary circulation. The caliper of pulmonary artery lumina decreases as smooth muscle and arterial walls contract. Contraction increases pulmonary artery pressure. Caliper increases as these muscles relax, decreasing blood pressure. Contraction or vasoconstriction and relaxation, vasodilation, primarily occurs in the response to local humoral conditions, even though the pulmonary circulation is innervated by the autonomic nervous system and the systemic circulation. The most important cause of pulmonary artery pressure or pulmonary artery constriction is a low alveolar partial pressure of oxygen, that PaO2. Vasoconstriction caused by alveolar and pulmonary venous hypoxia, often termed hypoxic pulmonary vasoconstriction, results from an increase in intracellular calcium levels in vascular smooth muscle cells in response to low oxygen concentration and the presence of charged oxygen molecules called oxygen-free radicals. It can affect only one segment of the lung, such as one lobe that is obstructed, and decreasing its PaO2, or the entire lung. If only one segment of the lung is involved, the arterioles to that segment constrict, shunting blood to other, well-ventilated portions of the lung. This reflex improves the lung's efficiency by better matching ventilation and perfusion. If all segments of the lung are affected, vasoconstriction occurs throughout the pulmonary vasculature, and pulmonary hypertension or elevated pulmonary artery pressure can result. The pulmonary vasoconstriction caused by low PaO2 values is reversible if the PaO2 level is corrected. Chronic alveolar hypoxia can result in inflammation and structural remodeling in pulmonary arterioles, causing permanent pulmonary artery hypertension, and that eventually would lead to your right-sided heart failure. Something to remember. Acidemia also causes pulmonary artery constriction. If acidemia is corrected, the vasoconstriction is reversed. An elevated PaCO2 value without a drop of pH does not cause pulmonary artery constriction. Other biochemical factors that affect the caliber of vessels in the pulmonary circulation include histamine, prostaglandins, endothelin, serotonin, nitric oxide, and bradykinin. So, the chest wall and your pleura. The chest wall, the skin, ribs, and intercostal muscles protect the lungs from injury. The intercostal muscles of the chest wall, in conjunction with the diaphragm, perform the muscular work of breathing. The thoracic cavity is contained by the chest wall and encases the lungs. A serous membrane called the pleura adheres firmly to the lungs. It then folds over itself and attaches firmly to the chest wall. The membrane covering the lungs is the visceral pleura. The lining in the thoracic cavity is the parietal pleura. The area between the two pleura is called the pleural space or pleural cavity. Normally, only a thin layer of fluid secreted by the pleura or pleural fluid fills the pleural space. About 18 ml of fluid is in the pleural space with a pH of about 7.6. A few cells, about 1 gram per deciliter of protein, and glucose and electrolyte concentrations that approximate those of the plasma. This lubricates the pleural space, allowing the two layers to slide over each other without separating. The pressure in the pleural space is usually negative at a subatmospheric level of -4 to -10 milligrams of mercury. Here we're taking a look at the thoracic chest cavity and its related structures. The thoracic cavity is divided into 3 subdivisions: left and right pleural division and the mediastinum, by a partition formed by a serous membrane called the pleura. So, functions of the pulmonary system. Again, the pulmonary system, it ventilates the alveoli, it diffuses gases into and out of the blood, and it perfuses the lungs so that the organs and tissue of the body receive the blood that is rich in oxygen and low in carbon dioxide. Each component of the pulmonary system contributes to one or more of these functions. So, the functional components of the respiratory system. The central nervous system responds to a neurochemical stimulation of ventilation and sends signals to the chest wall's musculature. The response of the respiratory system to these impulses is influenced by several factors that affect the mechanism of breathing and therefore affect the adequacy of ventilation. Gas transport between the alveoli and the pulmonary capillaries, or capillary blood, depends on the variety of physical and chemical activities. The control of the pulmonary circulation plays a role in the appropriate distribution of blood flow. Knowing which um, muscles are affected by that neurochemical control can help aid in knowing which portions will help with your breathing. Ventilation is the mechanical movement, gas or air into and out of the lungs. Ventilation is often misnamed respiration, which is actually the exchange of oxygen and carbon dioxide during cellular metabolism. The ventilatory rate, or respiratory rate, is the number of times gas is inspired and expired per minute. The amount of effective ventilation is caused by the multiplying the ventilatory rate, your breaths per minute, by the volume of the air per breath, your liters per breath, your tidal volume. This is termed the minute volume, or minute ventilation, and is expressed in liters per minute. Carbon dioxide, the gaseous form of carbonic acid, your H2CO3, is a product of cellular metabolism. The lung eliminates about 10,000 milliequivalents of carbonic acid per day in the form of CO2, which is produced at a rate of approximately 200 ml per minute. CO2 elimination is necessary to maintain a normal partial pressure of arterial CO2, or PaCO2, of 40 milligrams of mercury, and normal acid-base balance. The adequacy of alveolar ventilation cannot be accurately determined by observation of ventilatory rate, pattern, or effort. If a healthcare professional needs to determine the adequacy of ventilation, an ABG analysis must be performed to measure PaCO2. Neurochemical control of ventilation. Breathing is usually involuntary because homeostatic changes in the ventilatory rate and volume are adjusted automatically by the nervous system to maintain normal gas exchange. Voluntary breathing is necessary for talking, singing, laughing, and holding one's breath. The respiratory center in the brainstem controls respiration by transmitting impulses to the respiratory muscles, causing them to contract and relax. The medullary rhythmicity area is composed of interconnected groups of neurons located bilaterally in the brainstem. The dorsal respiratory group, or DRG, and the ventral respiratory group, VRG. The basic automatic rhythm of respiration is set by the ventral respiratory group, a cluster of inspiratory nerve cells located in the medulla that sends efferent impulses to the diaphragm and inspiratory intercostal muscles. The dorsal respiratory group receives different impulses from the peripheral chemoreceptors in the carotid and aortic bodies, from mechanical, neural, and chemical stimuli, and form receptors in the lungs, and it alters breathing patterns to restore normal blood gases. The pontine receptor respiratory center and apneustic center, situated in the pons, do not generate primary rhythm, but rather act as modifiers of the rhythm established by the medullary rhythmicity area. Breathing can be modified by the input from the cortex, the limbic system, and the hypothalamus, and the pattern of breathing can be influenced by emotion and by disease. Here we're taking a look of all the different ways your body may react to stimuli. The neurochemical respiratory control system. Again, it would not hurt to know which muscles can get affected. Your chemoreceptors. Chemoreceptors monitor your pH, your PaCO2, and your PaO2. Central chemoreceptors monitor arterial blood indirectly by sensing changes in the pH of cerebrospinal fluid, your CSF. They are located near the respiratory center and are sensitive to hydrogen ion concentration in brain interstitial fluid and CSF. The pH, or concentration of hydrogen ions in the CSF, reflects arterial pH because CO2 in arterial blood diffuses across the blood-brainstem barrier. the capillary wall that separates blood from the cells of the central nervous system, into the CSF until the partial pressure of CO2 is equal on both sides. CO2 that has entered the CSF combines with water to form carbonic acid, which subsequently dissociates into hydrogen ions that are capable of stimulating the central chemoreceptors. In this way, PaCO2 regulates ventilation through its effect on pH of that CSF. If alveolar ventilation is inadequate, your PaCO2 increases. That CO2 diffuses across the blood-brain barrier until the pCO2 values in the blood and the CSF reaches equilibrium. Because the central chemoreceptors sense the resulting decrease in pH with an increase in hydrogen ion concentration, they stimulate the respiratory centers to increase the depth and the rate of ventilation. Increased ventilation causes a PaCO2 of arterial blood to decrease below that of the CSF, and CO2 diffuses out of the CSF, returning its pH back to normal. The central chemoreceptors are sensitive to very small changes in the pH of CSF and are able to maintain a normal PaCO2 level under many different conditions, including strenuous exercise. If inadequate ventilation or hypoventilation is a long-term condition, such as chronic obstructive pulmonary disease, these receptors become insensitive to small changes in PaCO2, and the regulation of ventilation begins to become poor. In addition, prolonged increase in PaCO2 results in renal compensation through a bicarbonate retention. The bicarbonate gradually diffuses into the CSF, where it normalizes the pH and negates the effect of the ventilatory drive. Your peripheral chemoreceptors are located in aortic bodies, the aortic arch, and carotid bodies at the bifurcation of the carotids near the baroreceptors. They are somewhat sensitive to the changes in PaCO2 and pH, but are primarily sensitive to oxygen levels in arterial blood, your PaO2. As PaO2 and pH decrease, peripheral chemoreceptors, particularly in the carotid bodies, send signals to the respiratory center to increase its ventilation. However, if the PaO2 must drop below the normal, which is approximately 60 milligrams of mercury, before the peripheral chemoreceptors have much influence on ventilation, meaning it's not exactly the fastest response. If PaCO2 is elevated as well, ventilation increases much more than it would in response to either abnormality alone. The peripheral chemoreceptor become the major stimulus to ventilation when the central chemoreceptors are reset by chronic hypoventilation. So your lung innervation. 3 types of lung sensory receptors send impulses from the lungs to the dorsal respiratory group. Your irritant receptors are found in the epithelium of the conducting airway. They're sensitive to noxious aerosols like vapors, gases, and particulate matter, such as your dust, which causes them to initiate the coughing reflex. When stimulated, the irritant receptor also causes bronchoconstriction and increased ventilatory rate. These receptors are located primarily at the proximal layer or larger airways and are nearly absent in the distal airways. Thus, it is possible for secretions to accumulate in the distal respiratory tree without initiating a cough. Your stretch receptors, or slow adapting receptors, are located in the smooth muscles of the airways and are sensitive to increases in the size or volume of the lungs. They decrease ventilatory rate and volume when stimulated. An occurrence sometimes referred to as the Herring-Boyer expiratory reflex. This reflex is active in newborns and assists with ventilation. In adults, this reflex is active only at high tidal volumes, such as with exercise and mechanical ventilation, and may play a role in protecting against excess lung inflation. And then your pulmonary C fiber receptors, previously known as J receptors, Your juxtapulmonary capillary receptors are located near the capillaries in the alveolar septa and in other airway locations as nociceptors. They are sensitive to increased pulmonary capillary pressure, which stimulates them to initiate rapid, shallow breathing, your laryngeal constriction on expiration, and mucus secretion, hypotension, and bradycardia. They may be associated with a sensation of dyspnea. The lung is innervated by the autonomic nervous system. Fibers of the sympathetic division of the autonomic nervous system in the lungs branch from the upper thoracic and cervical ganglia of the spinal cord. Fibers of the parasympathetic division of the ANS travel in the vagus nerve to the lung. The parasympathetic and sympathetic division of the ANS control airway caliber by stimulating bronchial smooth muscle to contract or relax. The parasympathetic receptors cause smooth muscles to contract, whereas sympathetic receptors cause it to relax. Bronchial smooth muscle tone depends on equilibrium, that is, equal stimulation of contraction and relaxation. The parasympathetic division of the ANS is the main controller of the airway caliber under normal conditions. Constriction occurs if the irritant receptors in the airway epithelium are stimulated by irritants in inspired air, by inflammatory mediators, again that histamine, serotonin, and prostaglandin, also by many drugs, and by humoral substances. The mechanics of breathing. The mechanical aspect of inspiration and expiration are known collectively as the mechanics of breathing and involve major and accessory muscles of inspiration and expiration, elastic properties of the lungs and chest wall, and resistance to airflow through the conducting airways. Alterations in any of these properties increases the work of breathing, and thus the metabolic energy that must be exerted to achieve adequate ventilation and oxygenation of the blood. So what are our major and accessory muscles? The major muscles of inspiration are the diaphragm and the external intercostal muscles, the muscles between the ribs. The diaphragm is the primary muscle of ventilation. Its dome shape and separates the abdominal and thoracic cavity. It has multiple intrathoracic attachments and is innervated by the paired phrenic nerves. When the diaphragm contracts, it flattens downward, increasing the volume of the thoracic cavity, and creates a negative pressure that draws gas into the lungs through the upper airways and trachea. Contraction of the external intercostal muscles elevates the anterior portion of the ribs. This increases the volume of the thoracic cavity by increasing its front to back, your anterior posterior diameter. Although the external intercostal muscles may contract during quiet breathing, inspiration at rest usually is assisted by the diaphragm only. So here we're taking a look at the scalenus muscle, your intercostal muscles, your sternocleidomastoid, your pectoralis minor, your serratus anterior, your rectus abdominis, and then again your intercostal muscles, your serratus posterior superior in the back, serratus posterior interior, your diaphragm, and your transversus thoracis. So these are your muscles for ventilation. An anterior view in the front, a posterior view in the back. Again, these are good to know as some will show up on quizzes. The accessory muscles of inspiration are the sternocleidomastoid and the scalene muscles. Like the external intercostal muscles, these muscles enlarge the thorax by increasing that anterior-posterior diameter. The accessory muscles of inspiration assist inspiration when the minute volume, the volume of air inspired and expired per minute, is very high, such as during strenuous exercise and when the work of breathing is increased because of disease. The accessory muscles do not increase the volume of the thorax as efficiently as the diaphragm would. There are no major muscles of expiration because normal relaxed expiration is passive and requires no muscular effort. The accessory muscle of expiration, the abdominal and intercostal or internal intercostal muscles, assist expiration when your minute volume is high, when the person coughs, and when the airway obstruction is present. When the abdominal muscles contract, intra-abdominal pressure increases, pushing up that diaphragm and decreasing the volume of the thorax. The internal intercostal muscles pull down the anterior ribs, thereby decreasing that anterior-posterior diameter of the thorax. So that comes to alveolar surface tension. Surface tension occurs at any gas-liquid interface and refers to the tendency for liquid molecules that are exposed to air to adhere to one another. This phenomenon can be seen in the way liquids bead when splashed onto waterproof surface, decreasing the surface area exposed to the air. Within the sphere of the alveolus, surface tension tends to make expansion difficult, according to the law of Laplace. The pressure P required to inflate a sphere is equal to 2 times the surface tension, so 2T divided by the radius of the sphere. As the radius of the sphere becomes smaller, more and more pressure is required to inflate it. If the alveoli were lined with a water-like fluid, taking breaths would be extremely difficult. Alveolar ventilation or distension is made possible by surfactant, which lowers the surface tension by coating the air-liquid interface in the alveoli. Surfactant, that lipoprotein, produces a type 2 alveolar cell. Includes 2 groups of surfactant proteins. One group is your SPB and SPC. Consists of small hydrophobic molecules that have a detergent-like effect that separate liquid molecules, thereby decreasing alveolar surface tension. The second group, your SPA and SPD, consists of large hydrophilic molecules called collectants, pattern recognition molecules, that are capable of inhibiting foreign pathogens. Surfactant lines the alveolar side of the alveolar capillary membrane and, in effect, reverses the law of Laplace. As the radius of the surfactant-lined sphere of the alveolus becomes smaller, the surface tension decreases, and as the radius grows larger, the surface tension increases. This occurs because the surfactant molecules have much weaker intermolecular attraction than do liquid molecules. The surfactant molecules occupy most of the air-fluid interface and disrupt the intermolecular forces that tend to collapse the alveoli. Therefore, the alveoli are much easier to inflate at low lung volumes after expiration than at high volumes after inspiration. The decrease in surface tension caused by surfactant also is responsible for keeping the alveoli free of fluid. If surfactant production is disrupted or surfactant is not produced in inadequate quantities, such as in premature babies, alveolar surface tension increases, causing alveolar collapse, decreased lung expansion, increased work of breathing, and severe gas exchange abnormalities. Your elastic properties of the lung and chest wall. The lung and chest wall have elastic properties that permit expansion during inspiration and return to resting volume during expiration. The elasticity of the lungs is caused both by elastin fibers in the alveolar walls and in the surrounding small airways and pulmonary capillaries, and by surface tension at the alveolar air-liquid interface. The elasticity of the chest wall is supported by the configuration of its bones and musculature. Elastic recoil is the tendency of the lungs to return to the resting state after inspiration. Normal elastic recoil permits passive expiration, eliminating the need for major muscles of expiration. Passive elastic recoil may be insufficient during labored breathing, that high minute volume, in which case the accessory muscles of the expiration may be needed. The accessory muscles are used if disease compromises elastic recoil, such as near emphysema, or blocks the conducting airways. Normal elastic recoil depends on the equilibrium between opposing forces of recoil in the lungs and the chest wall. Under normal conditions, the chest wall tends to recoil by expanding outward when the sternum is split to open the thoracic cavity. The chest wall moves outward laterally. The tendency of the chest wall to recoil by expanding is balanced by the tendency of the lungs to recoil or collapse inward around the hila. This reaction is caused by elastic recoil and surface tension in the alveoli. The opposing forces of the chest wall and lung create, in part, the small negative intrapleural pressure. Balance between the outward recoil of the chest wall and the inward recoil of the lungs occurs at the resting level, at the end of expiration. During inspiration, the diaphragm and the intercostal muscles contract, which air flows into the lungs, and the chest wall expands. Muscular effort is needed to overcome the resistance of the lungs to expansion. During expiration, the muscles relax, and the elastic recoil of the lungs causes the thorax to decrease in volume until, once again, balance between the chest wall and the lung recoil forces is reached, triggering inspiration again. So here we're taking a quick peek. The interaction of forces during inspiration and expiration. Figure A, outward recoil of the chest wall equals inward recoil of the lungs at the end of expiration. Figure B, during inspiration, contraction of the respiratory muscles assisted by the chest wall recoil overcomes the tendency of the lungs to recoil. Figure C, at the end of inspiration, respiratory muscles contract, maintains lung expansion. And then figure D, during expiration, the respiratory muscles begin to relax, allowing elastic recoil of the lungs to deflate the lungs. And that would trigger figuration A again. The outward recoil of the chest wall equals inward recoil of the lungs, and at the end of expiration. Compliance is the measure of the lung and chest wall distensibility. It represents the relative ease with which the structures can be stretched. Compliance is therefore the reciprocal of elasticity. Compliance is determined by the alveolar surface tension and the elastic recoil of the lung and chest wall. It can be measured with the following formula, where C equals compliance in liters per centimeter of water. Your change in V would be volume change, usually your tidal volume, and your change in P, your pressure change, your airway or pleural pressure in centimeters of water. Increased compliance indicates that the lungs or chest wall is abnormally easy to inflate and has lost some of that elastic recoil. A decrease in compliance indicates that the lungs or chest wall is abnormally stiff or difficult to inflate. Compliance increases with normal aging and with disorders such as emphysema. It decreases in individuals with acute respiratory distress syndrome, pneumonias, pulmonary edema, and fibrosis. Airway resistance. Airway resistance, which is similar to resistance to blood flow, is determined by the length, radius, and cross-sectional areas of the airways, and by the density, viscosity, and velocity of the gas. It's Poiseuille's law. I probably butchered that name. Resistance is computed by dividing the pressure P by the rate of flow F or R, your Ohm's law, and can easily be measured in the pulmonary function lab. Airway resistance is normally very low, 1/2 to 2/3 of total airway resistance occurs in the nose. The next highest resistance is the oropharynx and the larynx. Airway resistance also is affected by the diameter of the airways, There's very little resistance in the conducting areas of the lungs because of their large cross-sectional area. Bronchodilation, which decreases resistance to the airflow, is caused by a beta-2 adrenergic receptor stimulation. Bronchoconstriction, which increases airway resistance, can be caused by stimulation of the parasympathetic receptors in the bronchial smooth muscle and by numerous irritants and inflammatory mediators. Airway resistance also can be increased by edema of the bronchial mucosa and by airway obstruction, such as mucus, tumors, and foreign bodies. Pulmonary function tests measure lung volumes and flow rates and can be used to diagnose different types of lung disease. Your work of breathing is determined by the muscular effort and therefore oxygen and energy required for ventilation. The work of breathing is normally very low, but may increase considerably in disease states that disrupt the equilibrium between forces exerted by the lungs and the chest wall. More muscular effort is required when lung compliance is decreased, your pulmonary edema, chest wall compliance is decreased, and spinal deformity or obesity, or airway is obstructed by bronchospasm and mucus plugging in your asthma and bronchitis. An increase in work of breathing can result in a marked increase in oxygen consumption and an inability to maintain adequate ventilation. So that leads us to our gas transportation. The delivery of oxygen to the cells of the body requires 4 steps: ventilation of the lungs, diffusion of oxygen from the alveoli into the capillary blood, perfusion of the systemic capillaries with oxygenated blood, and diffusion of oxygen from the systemic capillaries to its cells. The removal of CO2 also has 4 steps and is the reverse order of oxygen: the diffusion of CO2 from the cells into the systemic capillaries, perfusion of the pulmonary capillary bed by venous blood, your diffusion of CO2 into the alveoli, and the removal of CO2 from the lung via expiration. If any of these gas transport is impaired by the respiratory or cardiovascular disorder, gas exchange at the cellular level is compromised. So your measurements of gas pressure. The properties of air once it enters the body are determined by interactions among gas molecules at a given temperature. The millions of gas molecules are moving randomly, colliding with each other and the wall of the space in which they are contained. These collisions exert pressure. If the same number of gas molecules is contained in a small and a large container, the pressure is greater in the small container because the collision occurs in a smaller space. Heat increases the speed of molecules, which increases the number of collisions and therefore the pressure. So here you have a relationship between the gas molecules and the pressure exerted by the gas in the enclosed space. So in figure A, theoretically, 10 molecules of the same gas exert a total pressure of 10 milligrams of mercury within the space. In figure B, the number of molecules is increased to 20, so the total pressure would be 20 milligrams of mercury. In figure C, if there's a different gas in the space, the gas exerts a partial pressure. There's a partial pressure of nitrogen in the yellow, which is 18, and the partial pressure of oxygen, the blue, which is 6. So the total pressure would be 24 because it's all combined. Your barometric pressure, or atmospheric pressure, is the pressure exerted by gas molecules in air at specific altitudes. At sea level, barometric pressure is 760 milligrams of mercury. This number is the sum of pressure exerted by each gas in the air at sea level. The portion of the total pressure exerted by any individual gas is its partial pressure. So at sea level, the air is made of 20.9%, nitrogen is 78.1%, and a few other trace gases. So the partial pressure of oxygen is equal to the percentage of oxygen in the air times the total barometric pressure. So your 760 goes down to 159 because 760 times 0.209 equals 58.84. And you'll just round that up a little bit. Symbols used in the measurement of gas, pressure, and pulmonary ventilation are shown here at this next table. And there are a lot. But things we should know. So, the amount of water vapor contained in a gas mixture is determined by the temperature of the gas and is unrelated by your barometric pressure. Gas that enters the lungs becomes saturated with water vapors and gets humidified as it passes through the upper airway. At body temperature 37 degrees Celsius, water vapor exerts a pressure of 47 milligrams of mercury, regardless of the barometric pressure. The partial pressure of water vapor must be subtracted from the barometric pressure before the partial pressure of other gases in the mixture can be determined. So, in air saturated with water at sea level, the partial pressure of oxygen is therefore your 760 minus 47, but still times that 0.209. So it comes down to 149. All pressure and volume measurements made in pulmonary function laboratories specify the temperature and the humidity of the gas at the time of measurement. Effective gas exchange depends on an approximately even distribution of gas, your ventilation, and blood, your perfusion, in all portions of the lungs. The lungs are suspended from the highlight of the thoracic cavity. When the individual is in an upright position, sitting or standing, gravity pulls the lungs down towards the diaphragm and creates a greater negative pleural pressure in the upper portions, or apexes of the lungs, than at the base. The alveoli in the apexes also contain a greater residual volume of gas and are larger and less numerous than those in the lower portions. Surface tension increases as the alveoli become larger. The combination of greater negative pleural pressure and higher surface tension in the larger alveoli in the apex of the lungs makes them more difficult to inflate. They're less compliant and less distensible than the smaller alveoli in the lower portions of the lungs. Therefore, during ventilation, most of the tidal volume is distributed to the bases of the lungs, where compliance is greater. The heart pumps against gravity to perfuse the pulmonary circulation. As blood is pumped into the lungs, or into the lung apexes of the sitting or standing individual, some blood pressure is dissipated in overcoming gravity. As a result, blood pressure at the apexes is lower than that at the bases, because greater pressure causes greater perfusion. The bases of the lungs are better perfused than the apexes. Thus, ventilation and perfusion are greatest in the same lung portions. The lower lobes, ventilation, and perfusion depend on the body position. If a standing individual assumes a supine or side-lying position, the areas of the lungs that are then most dependent become best ventilated and perfused. So here you see the difference in how lung perfusion would work, whether you're sitting and standing, side-lying, or supine. It's all about that blood pressure. Distribution of perfusion in the pulmonary circulation also is affected by alveolar pressure, that gas pressure in the alveoli. The pulmonary capillary bed differs from the systemic capillary bed in that it is surrounded by gas-containing alveoli. If the gas pressure in the alveoli exceeds the blood pressure in the capillary collapses and flow ceases. This occurs in portions of the lung where blood pressure is lowest and alveolar gas pressure is greatest, that is, in the apex of the lung. The lungs are divided into three zones on the basis of the combination of factors affecting pulmonary blood flow: alveolar pressure and the forces of gravity, arterial blood pressure, and venous blood pressure determine the distribution of perfusion. Taking a peek here, gravity and alveolar pressure. The effects of gravity and alveolar pressure on pulmonary blood flow in the three lung zones. In zone 1, alveolar pressure, your PA is greater than the blood, arterial, and venous pressure, and no blood flow occurs. In zone 2, however, arterial blood pressure, PA, exceeds alveolar pressure, but alveolar pressure exceeds venous pressure. Your blood flow occurs in this zone, but the alveolar pressure compresses the venules. In zone 3, both arterial and venous pressure are greater than alveolar pressure, and blood flow fluctuates depending on the difference between the arterial and venous pressure. So, in zone 1, alveolar pressure exceeds pulmonary, arterial, and venous pressure. The capillary bed collapses, and normal blood flow ceases. And zone 1 is a very small part of the lung at the apex. In zone 2, alveolar pressure is greater than the venous pressure, but not greater than the arterial pressure. So blood flows through zone 2, but is impeded to a certain extent by the alveolar pressure. Zone 2 is above the level of the left atrium. Zone 3, arterial and venous pressure are greater than the alveolar pressure, and blood flow is not affected by any kind of alveolar pressure. Zone 3 is at the base of the lungs. Blood flows through the pulmonary capillary bed, increases in regular increments from the apex to the base. So, although the blood flow and ventilation are greater at the base of the lungs than at the apexes, they're not perfectly matched in any zone. Perfusion exceeds ventilation in the bases, and ventilation exceeds perfusion in the apex of the lungs. That relationship between ventilation and perfusion is expressed in a ratio called the ventilation-perfusion ratio, that VQ. The normal VQ is 0.8. This is the amount by which the perfusion exceeds ventilation under normal conditions. So again, your ventilation over perfusion. Your oxygen transport. Approximately 1 liter of oxygen is transported to the cells of the body each minute. Oxygen is transported in the blood in two forms. The small amount dissolves in plasma, and the remainder binds to hemoglobin molecules. Without hemoglobin, oxygen would not reach the cells in amounts sufficient to maintain normal metabolic function. We talked about hemoglobin in chapter 28. Diffusion across the alveolar capillary membrane. The alveolar capillary membrane is the ideal medium for oxygen diffusion because it has a large total surface area. Again, it's that 70 to 100 meters squared, and is very thin. That's 0.5 nanometer. In addition to partial pressure of oxygen molecules, your PO2 is much greater in alveolar gas than in capillary blood, a condition that promotes rapid diffusion down the concentration gradient from the alveolus into the capillary. The amount of oxygen in the alveola, your PaO2, depends on the amount of oxygen in the inspired air and the amount of oxygen that remains in the alveoli and tracheobronchial tree, that physiologic dead space, between breaths. The PaO2 can be estimated by using an alveolar gas equation. So your PaO2 equals your PiO2 minus your PaCO2 divided by 0.8, your respiratory quotient, where PiO2 is the inspired partial pressure of oxygen, which can be calculated by measuring your barometric pressure minus the 47 of vapor pressure of water times your FiO2. That's 0.209 or 0.021 or 0.21 if you round up. The PaO2 value is approximately 104 milligrams of mercury at sea level with relaxed breathing. Therefore, a pressure gradient of approximately 60 milligrams of mercury facilitates the diffusion of oxygen from the alveolus into the capillary. Different values of PaO2 can be calculated if there are changes in inspired oxygen content or your PaCO2, which are common occurrences in clinical settings. So here you have a diagram of all the different measures. Partial pressure of respiratory gases in normal respiration. These are average values. The values of PaO2, PCO2, and PN2 fluctuate from breath to breath. Your CO2 carbon dioxide, your O2 is oxygen, your PCO2 is your partial pressure of carbon dioxide, your pH2O is your partial pressure of water, your PN2 is partial pressure of nitrogen, and PO2 is partial pressure of oxygen. So there is quite a bit of fluctuation per breath. Blood remains in the pulmonary capillary for about 0.75 seconds, but only 0.25 seconds is required for oxygen concentration to equilibrate across the alveolar capillary membrane. Therefore, oxygen has ample time to diffuse into the blood. Even during increased cardiac output, which speeds blood flow, shortening the time of blood remaining in the capillaries. Determinants of arterial oxygenation. As oxygen diffuses across the alveolar capillary membrane, it dissolves in the plasma, where it exerts pressure, the partial pressure of oxygen in the arterial blood, your PaO2. As that partial pressure of oxygen increases, oxygen moves from the plasma into the red blood cells, your erythrocytes, and binds with hemoglobin molecules. Oxygen continues to bind with hemoglobin until the hemoglobin binding sites are filled or saturated. Oxygen then continues to diffuse across the alveolar capillary membrane until the PaO2 and PaO2 equilibrate, eliminating the pressure gradient across the alveolar capillary membrane. At this point, diffusion ceases. The majority, about 97% of oxygen that enters the blood is bound to hemoglobin, the remaining 3% to plasma. The PaO2 can be measured in the blood by obtaining an arterial blood gas measurement. The oxygen saturation is the percentage of the available hemoglobin that is bound to oxygen and can be measured using a device called an oximeter. The total amount of oxygen carried in the blood is the oxygen content and is measured in milliliters per deciliter of blood. It is the combined value of the oxygen in oxygen saturation hemoglobin and the oxygen dissolved in the blood. To calculate total arterial oxygen content, one must know 1. The hemoglobin concentration, or the amount of hemoglobin that is available to bind with oxygen, 2. The oxygen saturation, or the percentage of available hemoglobin that is bound to oxygen, and 3. The partial pressure of oxygen. The maximum amount of oxygen that is, or the maximum amount of oxygen that can be transported by hemoglobin is 1.34 milliliters per gram. The amount of oxygen that can be physically dissolved in blood is only 0.003 ml per deciliter per milligrams of mercury. With the specific values known, the oxygen content of arterial blood can be calculated. To calculate the oxygen content of venous blood, the partial pressure is mixed with venous blood, your PVO2, and venous oxygen saturation, your SVO2, or substituted for the arterial values in the basic formula. Normal venous oxygen content is about 15 to 16 milliliters per deciliter. Because hemoglobin transports all but a small fraction of oxygen carried in arterial blood, increases in hemoglobin concentration affect the oxygen content of the blood. Decreases in hemoglobin concentration below the normal value of 15 ml per deciliter of blood reduces oxygen content. Increases in hemoglobin concentration may increase oxygen content, minimizing the effect of impaired gas exchange. In fact, increased hemoglobin concentration is a major compensatory mechanism in pulmonary disease that impairs gas exchange. For this reason, measurements of hemoglobin concentration is important in assessing individuals with pulmonary disease. If cardiovascular function is normal, the body's initial response to low oxygen content is to acquire cardiac output. In individuals who have cardiovascular disease, this compensatory mechanism is ineffective. An elevated hemoglobin level can be compensated for an inability to accelerate cardiac output, making increased hemoglobin concentrations an even more important compensatory mechanism. So that leads us to our oxyhemoglobin association and dissociation. Your oxyhemoglobin, or HbO2, forms when hemoglobin molecules binds with oxygen. Binding occurs in the lungs and is called oxyhemoglobin association or hemoglobin saturation with oxygen, your SaO2. The reverse process, where oxygen is released from that hemoglobin, occurs in the body tissues at a cellular level and is called hemoglobin desaturation. When hemoglobin saturation and desaturation are plotted on a graph, the result is a distinct S-shaped curve known as the oxyhemoglobin dissociation curve. For PaO2 values less than 60 milligrams of mercury, oxygen is readily unloaded in peripheral tissue and hemoglobin's affinity for oxygen diminishes, reflecting the steep part of the curve. For PaO2 values greater than 60 milligrams of mercury, the curve becomes relatively flat, reflecting the maximum saturation of hemoglobin with oxygen in the lungs. So here we're taking a look at that curve. Your oxyhemoglobin dissociation curve, the horizontal or the flat segment of the curve at the top of the graph, is the arterial or associated portion, or that of the curve where oxygen is bound to hemoglobin, and that usually occurs in the lungs. This portion of the curve is flat because partial pressure changes of oxygen between 60 and 100 milligrams of mercury do not significantly alter the percentage of saturation of that hemoglobin with oxygen and allow adequate hemoglobin saturation at a variety of altitudes. If that relationship between SaO2 and PaO2 was linear in a downward sloping straight line instead of flat, there would be an inadequate saturation of hemoglobin with oxygen. The steep part of the oxyhemoglobin dissociation curve represents the rapid dissociation of oxygen from hemoglobin that occurs in the tissues. During this phase, there is a rapid diffusion of oxygen from the blood into the tissues. The P50 is where the PaO2 is at which hemoglobin is 50% saturated, normally 26.6 milligrams of mercury. A lower than 50 represents an increased affinity of hemoglobin for O2. A high P50 is seen with a decreased affinity. Note that the variation from the normal is associated with a decreased or increased availability of oxygen to the tissues. The shaded areas show the entire oxyhemoglobin dissociation curve under the same circumstances. Your 2,3-DPG or 2,3-diphosphoglycerate. So, several factors can change the relationship between PaO2 and SaO2 and influence that hemoglobin affinity for oxygen. Again, at 26.6 milligrams of mercury, that PaO2, hemoglobin is 50% saturated, known as P50. An increase in P50 value shifts the curve to the right, indicating hemoglobin's affinity or decreased affinity for oxygen, and an increase in the ease at which oxyhemoglobin dissociates, and oxygen moves into the cells. A larger partial pressure is necessary to maintain 50% oxygen saturation. A shift to the left depicts hemoglobin's increased affinity for oxygen, which promotes association in the lungs and inhibits dissociation in the tissues. A lower partial pressure is necessary to maintain 50% oxygen saturation. The oxyhemoglobin dissociation curve is shifted to the right by acidosis and hypercapnia. In the tissues, increased levels of CO2 and hydrogen ions produced by metabolic activity decreases the affinity of hemoglobin for oxygen. Hyperthermia and increased levels of that 2,3-diphosphoglycerate also shift the curve to the right. The curve is shifted to the left by alkalosis, or hypocapnia, hypothermia, and low levels of that 2,3-diphosphoglycerate. In the lungs, as CO2 diffuses from the blood into the alveoli, the blood CO2 level is reduced and the affinity of hemoglobin for oxygen is increased. The shift in oxyhemoglobin dissociation curve caused by changes in CO2 and hydrogen ion concentration in the blood is called the Bohr effect. So it's actually really important to note how your oxygen will transfer faster into your tissues if the patient has hyperthermia, increased levels of that 2,3-diphosphate, or are acidotic or hypercapnic. However, you would notice that there is that decreased shift if you have alkalosis, hypocapnia, hypothermia, or low levels of that 2,3-DPG level. So keep that in mind. Your carbon dioxide transport is approximately 200 milliliters of CO2 and is produced by the tissues per minute at rest as a byproduct of cellular metabolism. This CO2 equilibrates with carbonic acid, your H2CO3, and must be eliminated continuously to prevent acidosis. The elimination of CO2 by the lungs plays an important role in regulation of that acid-base balance. CO2 is carried in the blood in three ways. It dissolves in plasma, it's transported as a bicarbonate, or it is combined with blood proteins to form carb-amino compounds. As CO2 diffuses out of the cells into the blood, it dissolves in the plasma. Approximately 10% of the total CO2 is in venous blood and 5% of the CO2 is in arterial blood and are carried dissolved into the plasma. As CO2 moves into the blood, it diffuses into the red blood cells. With the red blood cells, CO2, with the help of the enzyme carbonic anhydrase, combines with the water to form carbonic acid and then quickly dissociates into hydrogen and bicarb. As carbonic acid dissociates, the hydrogen binds to hemoglobin, where it is buffered, and the bicarb moves out of the red blood cells into the plasma. About 60% of CO2 in venous blood and 90% of CO2 in arterial blood are carried in the form of bicarbonate. The remainder combines with blood proteins, hemoglobin in particular, and form carb-amino compounds. Roughly 30% of the CO2 in venous blood and 5% of CO2 in arterial blood are carried as carb-amino compounds. So you'll see all three in plasma and blood, both venous and arterially. CO2 is 20 times more soluble than oxygen and diffuses quickly from the tissue cells into the blood. The amount of CO2 that is able to enter the blood is enhanced by diffusion of oxygen out of the blood and into the cells. Reduced hemoglobin is able to carry more CO2 than hemoglobin that is saturated with oxygen. Therefore, a drop in SaO2 at the tissue level increases the ability of hemoglobin to carry CO2 back to the lung. The diffusion gradient of CO2 in the lung is only approximately 6 milligrams of mercury, yet CO2 is so soluble in the alveolo-capillary membrane that the CO2 in the blood quickly diffuses into the alveoli, where it is removed from the lung with each expiration. Diffusion of CO2 in the lung is so efficient that diffusion defects and causes hypoxemia, but does not cause hypercapnia. The diffusion of CO2 out of the blood also is enhanced by oxygen binding with hemoglobin in the lung. As hemoglobin binds with oxygen, the amount of CO2 carried by the blood is decreased. Thus, in the tissue capillaries, O2 dissociation from hemoglobin facilitates the pickup of CO2, and the binding of O2 to hemoglobin in the lungs facilitates the release of CO2 from the blood. This effect of oxygen on CO2 transportation is called the Haldane effect. That leads us to our pulmonary function test. Several lab tests aid in the diagnosis and evaluation of pulmonary system abnormalities. Most of them are easy to perform in the hospitals and clinics. They provide valuable information about the possible cause of the respiratory abnormality and evaluate the progression or resolution of disease. Spirometry measurements and the volume of flow of an air inhaled and exhaled and is plotted against time, that flow rate, during different breathing maneuvers. Several different types of spirometry are available. The procedure produces a spirogram, which is a record of individual ventilation in relation to time. Clinically, the most important spirometric tests are the forced vital capacity, your FVC, and the forced expiratory volume in 1 second, your FEV1. These tests and other important measurements are described again in that earlier table. Spirometry enables clinicians to detect restrictive or obstructive deficits early in the course of disease. Restrictive lung disease restricts the lung's volume. The lungs are unable to expand normally, diminishing the amount of gas that can be inspired, and reduces the FVC. Obstructive lung disease affects the airflow. Airflow into and out of the lungs is obstructed, reducing FEV1. So, looking at our figure here, your pulmonary ventilation and lung capacity. A spirogram during normal quiet respirations, the atmosphere and lungs exchange about 500 ml of air. With a forcible inspiration, about 3,300 ml, more air can be inhaled. After normal inspiration and normal expiration, approximately 1,000 ml, more air can be forcibly expired. The vital capacity is the amount of air that can be forcibly expired after maximal inspiration, and indicates therefore the largest amount of air that can enter and leave the lungs during respiration. Your residual volume is the air that remains trapped in the alveoli. Figure B shows lung capacities. So here we have some symbols of ventilatory property measures. Your FVC, that forced vital capacity, your FEV1, your forced expiratory volume in 1 second. Then you have the measurement, that percentage of maximum inspired that is expired in 1 second is usually 80%. Your FEV3, that's the forced expiratory volume in 3 seconds. And then the measurement percentage of FVC that is expired in 3 seconds is usually 95%. And then you have that FEF 25 to 75%, that forced expiratory flow rate during the middle 50% of expiration, sometimes reported as a maximum mid-expiratory flow rate. Your MVV, your maximum voluntary ventilation, and your PEF, your peak expiratory flow. So lung capacities, such as vital capacity and total lung capacity, are always the sum of 2 or more volumes. Norms for volumes and capacities are based on age, sex, height, and are referred to as predictive values. Differences from predicted or changes from baseline values are taken into account in diagnosing and assessing respiratory disorders. Your pulmonary diffusing capacity is a measure of the rate of gas diffusion across the alveolo-capillary membrane. Oxygen, or more commonly carbon monoxide, is used to measure diffusing capacity. Carbon monoxide has a high affinity for hemoglobin and is not limited by pulmonary blood flow and therefore is an excellent measure of diffusion. However, it's poisonous. The measurement is made by determining how much carbon monoxide is taken up by the blood and dividing this amount by the pressure gradient across the alveolo-capillary membrane. Helium often is added into the gas mixture to obtain a simultaneous measurement of residual volume, function residual capacity, and total lung capacity, that RF, FRC, and TLC. Individuals are asked to perform ventilatory maneuvers similar to those used in spirometry testing. A decreased diffusing capacity can be the result of an abnormal ventilation-perfusion ratio, or an actual diffusion defect. For example, diffusing capacity is decreased in individuals with emphysema, where there is a decrease in surface area of the pulmonary capillaries available for gas exchange. Oximetry is commonly performed for individuals with suggested or diagnosed pulmonary disease. It indirectly measures hemoglobin oxygen saturation and can suggest that PaO2 is low. However, it is prone to inaccurate measurements, particularly in individuals with poor peripheral circulation, and does not measure the PaCO2 or pH of the blood. Capnography measures the amount of CO2 in expired air and thus estimates PaCO2. But this test is also prone to inaccuracy. Your ABG analysis is the direct measurement of pH and gas concentrations in arterial blood and provides more accurate and complete information about an individual's gas exchange and acid-base status. Acidosis, alkalosis, ventilatory alterations, and decreased PaO2 can be diagnosed accurately only via an ABG. A blood gas report may be divided into an acid-base ventilation portion and oxygenation portion. Oximetry can be used to monitor oxygen saturation once the arterial blood gas analysis has accurately measured the PaO2, but it does not measure your PaCO2 or pH. And then we have our acid-base measurements. Signs and symptoms of most respiratory abnormalities first appear when the system is stressed during exercise. Therefore, if pulmonary disease is suspected, the individual is elevated at rest and during exercise. Evaluation during exercise usually entails spirometry and oximetry to monitor hemoglobin saturation. The exercise usually consists of riding a stationary bike or walking on a treadmill. Exercise testing enables clinicians to detect early changes in respiratory function and thus begin treatment. Exercise tests also are used in planning and evaluating exercise and rehabilitation programs. Your thoracic imaging techniques are among the most common examinations for diagnosis and detection of pulmonary disease and tumors, and for evaluation of disease progression. Numerous techniques are available and include chest radiography, CT, MRI, positron emission tomography, and ultrasound. Pulmonary angiography is used for the detection of pulmonary emboli and pulmonary arteriovenous malformations. Techniques may be combined for the best evaluation of pulmonary structure and function. So aging and the pulmonary system, most knowledge about pulmonary structure and function is based on the norms for the middle-aged years. Less is known about the structure and function of the very young and in older adults, but a few normal physiologic changes are known to occur from birth to old age. An understanding of these changes is necessary to provide appropriate care and to differentiate between age-associated alterations and disease. Normal alterations in pulmonary system include the loss of elastic recoil, stiffening of the chest wall, and changes in gas exchange, and increases in flow resistance. These changes, which affect lung volumes, are gradual and usually without adverse consequence in healthy individuals. They are influenced by genetics, sociocultural factors, nutritional status, exercise, decreased immune function, respiratory disease, body size, sex, and race. Exposure to environmental toxins such as some respiratory tract infections, tobacco smoke, air pollution, and occupational dust also contribute to decreased lung function with age. The changes contribute to increased lung disease morbidity and mortality in the older adult, including chronic obstructive lung disease, your COPD, lung cancer, pulmonary fibrosis, and infection. During adulthood, as the age advances, the alveoli tend to lose alveolar wall tissues and capillaries. This process increases alveolar size and diminishes the alveolar surface area available for gas diffusion, and decreases airway support provided by normal lung tissue. These changes are similar to those seen in emphysema and change the elastic properties of the lungs. Chest wall compliance decreases with age because the ribs become ossified and the joints become stiffer. As a result, the chest wall loses some of its ability to expand. In addition, respiratory muscle strength and endurance decrease by up to 20% by the age of 70. These mechanical changes in the lung and chest wall, along with the structural changes in the alveoli, reduce the ventilatory capacity in older adults. Vital capacity decreases and residual volume increases. However, total lung capacity remains unchanged. These changes decrease ventilatory reserve and lead to a decreased ventilation-perfusion ratio. With advancing age, there is also increased immune dysregulation, asymptomatic low-grade inflammation, and increased risk of infection. Alterations in gas exchange are reflected by blood gas analysis with advancing age, pH, and PaCO2. Levels do not change much, even though it has been documented that the chemoreceptors become less sensitive to gas partial pressure with age. Older adults have a decreased compensatory response to hypercapnia and hypoxemia. However, the perception of dyspnea remains intact and is even enhanced. Your PaO2 declines with age as a result of structural and mechanical changes, such as loss of alveolar surface area, thickening of pleural septa, loss of lung elasticity, and increase in ventilation perfusion mismatch. The maximum PaO2 value in an older adult at sea level can be estimated by multiplying the person's age by 0.3 and subtracting the product from 100. So if your 80-year-old individual would have an estimated PaO2 of 76 milligrams of mercury. The decrease in PaO2 and the diminished ventilatory reserve in an older adult lead to a decrease in exercise tolerance, respiratory muscle strength, and endurance decreased with age. Furthermore, older adults are at greater risk for respiratory depression caused by medication. Changes in respiratory structure and function can vary considerably from person to person. However, changes are also affected by activity and fitness early in life. And that concludes our chapter here on the structure and function of the pulmonary system. We'll continue on next with the alterations of the pulmonary function in chapter 36. I'll see you then.